Hello everyone, on behalf of Wiley, welcome to our webinar today. Before we begin, I would like to address a couple of housekeeping items. First, we strongly encourage audience engagement, so please feel free to introduce yourself in the attendee chat and engage with each other. If you have questions, please put them in the chat and we'll do our best to address them during the live Q&A at the end of today's webinar. And if you're having issues seeing the broadcast, please try to refresh your web page or clear your cookies. We recommend using Google Chrome as your browser for the broadcast. So I would like to welcome Professor Adrian Liston, who is Professor of Pathology at the Department of Pathology at the University of Cambridge. Professor Liston's talk today is titled Regulatory T cells in the tissues, characteristics and importance of non-lymphoid tissue-resident T regulatory cells, in which he will discuss recent findings on FOXP3 plus regulatory T cells, or Tregs, in tissues, challenging the view of tissue-specific Tregs by presenting evidence for a single, broadly self-reactive Treg pool with implications for neuroinflammation treatment. This broadcast will be recorded and available to watch on demand at your convenience. Now, please welcome Professor Liston.
Thank you very much. It's a pleasure to be here today. So I'm going to discuss the population of tissue regulatory T cells, or Tregs, but also I want to take a particular focus of using in vivo multiplexed flow cytometry tools in order to study the tissue Treg population. And while a lot of our findings are really relevant to the regulatory T cell field, the tools that we use for in vivo multiplexing have a lot of broad utility across multiple different fields. So the population of regulatory T cells are well known to be able to control the balance between immunity and tolerance. We know that this population of FOXP3 positive Tregs are able to prevent any immune reaction, essentially. They can control autoimmunity, they can control anti-cancer responses, they control infection responses. And almost all of the studies that have been done on Tregs are based on this systemic population that is present in the spleen and lymph nodes or in the peripheral blood. However, there is a growing appreciation that there's also a population of regulatory T cells that are present inside essentially every organ. It's thought that they are required for local immune control inside these organs, although that's not always quite clear, and a lot of the reaction inside the organ may actually be controlled from the draining lymph node rather than from the organ inside. But beyond immune control, it looks like these tissue Tregs have important roles in the homeostasis of the tissues. In the gut Tregs can alter the microbiome. In the adipose resident Tregs can improve insulin sensitivity. Muscle Tregs can help muscle repair. Brain Tregs can aid oligodendrocyte differentiation, and so forth. So they have this set of really important functions that don't only dampen down immunity, but they also seem to promote repair and return to homeostasis after any type of damage or inflammation. Now, when we started working in this area, the model of the tissue Tregs was based on the tissue seeding and specialization model that people might know from tissue macrophages. And under this model, regulatory T cells are going from the periphery that have a distinct seeding event into multiple different tissues, and in those tissues they specialize, such that the Treg you see in the liver will be different from the Treg you see in the lung, and so forth. And then upon specialization, they have an indefinite residency. And so you have this long-term population that lives inside organs. So this is what I'll call the tissue seeding and specialization model. Now, we wanted to test this model by looking at multiple different tissues in detail simultaneously. And we first looked at whether or not the phenotypes that we see for tissue Tregs inside different tissues are actually that different. This experiment here is based on high-dimensional flow cytometry. The work is done by Oliver Burton, our flow cytometry guru. And what we've got here is mice where we have perfused the organs. We've collected 48 different colors, and we can also exclude any vascular contamination by giving an intravenous injection of anti-CD45 and then having that as an additional color. This is a 46 color spectral panel, and essentially we can phenotype at a single cell level through high dimensional flow cytometry the Tregs present across multiple different organs. Now you see four main phenotype blocks here. There are regulatory T cells that are in the blood, and these blood regulatory T cells are either naive or generically activated. The population of regulatory T cells in the lymphoid tissue are much more biased towards activated populations. Then you'll see the tissues associated with the gut have got a set of phenotypes that encompasses the generic activated population, but also has these unique populations that are neuropilin 1 negative, often associated with CD103. That's a very gut phenotype. And then finally, you've got the classical non-gut, non-lymphoid tissues. And here what we see is that the phenotype of the regulatory T cells across all of these different non-gut, non-lymphoid tissues basically looks the same. They're enriched in activated cells, but they're also in particular enriched in a population of activated cells that also express CD69, ST2, some of these classical residency markers. Now we've done similar work with sequencing studies, so both for single cell sequencing and bulk sequencing. I won't go through that in detail today, but essentially we can take regulatory T cells from different tissues and then look at how transcriptionally similar they are. And if you take a pair such as pancreatic Tregs and kidney Tregs, those populations are transcriptionally 98% identical. So these are very, very similar or identical cells, even though they are present in multiple different organs. We certainly do not see unique genes that are expressed in Tregs from one particular tissue, with the exception of the gut, where that population becomes more like 90% similar or 85% similar, rather than this sort of 98, 95% similar range. If you're interested in the data, this is all openly available, and it's also an interactive, easy to use viewer online. You can just use the QR code there. We go into multiple different phenotypes over different tissues, different ages, different microbiomes, and it's all available online. Now, the other aspect we want to work out is whether or not these populations are actually indefinite residents. So for this, we did a parabosis experiment. We could take CD45.1 and CD45.2 mice, so we can differentiate the cells based on this marker, CD45.1, CD45.2. If you surgically join those mice together, the vascular system joins up very quickly. And so now all of the cells that are in the blood are circulating and normalized. You've got this mixture of cells. We can then take down the mice at different time points post-parabiosis, look at multiple different organs, and try to calculate the rate at which regulatory T cells from the blood displace the population that is present inside the tissue. And what we find is that it's the population of activated cells that are activated regulatory T cells that are good at getting into the tissues. And most of these cells that enter from the blood have a very short life. They'll only spend a couple of days in the tissue and then they'll die or leave, while about half of them will transition into this population of CD69 positive regulatory T cells that can last longer in the tissue. It's important, though, that the length of time that they last is not indefinite. They are generally staying about 3 weeks in the tissue before they either die or dedifferentiate and leave. So this is the residency time calculated for the CD69 positive regulatory T cell population. So this is based on the cells that really have this residency phenotype. And it turns out that across multiple different organs, the average duration of staying is about 3 weeks. It is a bit longer in some tissues, so about 10 weeks in the adipose tissue or in the gut tissues, but it's really in this weeks to months range and not an indefinite dwell time like tissue-resident macrophages. So if we go back to this tissue seeding and specialization model, remember the key features of this model that has been assumed and does look like the valid model for tissue-resident macrophages are distinct seeding, unique specialization, and indefinite residency. Now, when it comes to regulatory T cells, this is not supported by the data. We don't see a distinct seeding event. We see that the cells are continually entering. We don't see unique specialization. There is specialization towards being in a tissue, but each tissue looks like it's turned on a very similar program. And we don't see indefinite residency because the parabosis data says that these cells can leave relatively quickly. So the model that we think is valid for regulatory T cells is a model where there are these pan-tissue cells. So there are regulatory T cells that are present in circulation. They're able to enter different tissues. When they enter a tissue, they turn on this tissue residency module. That module is more or less the same in multiple different tissues. They'll then percolate through that tissue for a number of weeks to months before they leave and go back into circulation. In fact, the same regulatory T cell under this model could enter multiple different tissues over the course of its lifetime. Now, we do have an exception here where the gut Treg population does look like it's got some unique features and some unique circulatory patterns. But for the other tissues, we believe the pan-tissue model explains more of the data than the seeding and specialization model. So if we want to test this model of pan-tissue resident regulatory T cells, we came up with 3 different hypotheses that we can use to test the model. Now, the first test is that the regulatory T cells in different tissues should share the T cell receptor. Now, T cell receptor is quite unique among genes because it is embedded in the germline in a pseudogene formation, and each lineage of T cells has to recombine that locus at the germline level. So it acts as a unique barcode for each clone of T cells. So if you are seeing the same barcode in multiple different tissues, you know that that single clone of T cells, or in our case, regulatory T cells, is actually able to enter multiple tissues at the same time, so that you can find that barcode across multiple tissues. So the way that we tested this was we extracted regulatory T cells from multiple different tissues, the blood, the pancreas, the kidney, the liver, and the gut. We then did single cell sequencing using the 10x VDJ sequencing protocol that allows us to get TCR alpha and TCR beta chains at a single cell level. And then we can look at how much sharing we see for regulatory T cells in one tissue compared to regulatory T cells in another tissue. So the data I'm showing here is a chord diagram where around the outer ring, we have the different tissues. The inner ring is 4 different mice per tissue. And then any of the lines that are connecting are T cell receptors that we're spotting in multiple different cells. Now, what you can see here is that the T cell receptors that we identify from gut regulatory T cells are shared between different mice in the gut, and there is some sharing from the gut to other tissues, but not a whole lot. This is in contrast to if you look at the liver regulatory T cells, where you see a lot of sharing between mouse to mouse of regulatory T cell TCRs in the liver, but you also see a lot of sharing with other organs like the kidney and the pancreas. In fact, we can calculate how much sharing that we see based on a normalization ratio. And if we do this, we see about 60 to 70% sharing between the kidney, the liver, and the pancreas, while the amount of sharing between the gut and those organs is more like 10%. So there's very strong sharing across the non-lymphoid, non-gut tissues. Now, the second part of this model is that we should be able to confer multi-tissue homing capacity on a regulatory T cell by giving it one of these T cell receptors. Now, this is because the T cell receptor, as well as acting as a barcode, it is what confers which antigen is recognized by the regulatory T cell. So if it recognizes an antigen present in multiple different tissues, it should be able to go into multiple different tissues. While if it's recognizing an antigen only present in one tissue, we might expect that TCR to confer homing only to one tissue. Now, there's a way that you can test the effect of T cell TCRs, and this is by creating a TCR transgenic. Now, under this system, what you would do is you would take a T cell, and then you would force that T cell to express the T cell receptor from one of these tissue Tregs. You've now got a T cell with this transgenic TCR that we have predefined because we've cloned the TCR and we've forced this cell to express that particular TCR. We can then measure the fate of that TCR, of that T cell, and we can say, did the transgenic TCR drive a Treg fate? Did that T cell become a Treg? And did it drive a monotissue or multi-tissue homing pattern? In other words, are the Tregs in one tissue or in multiple tissues? Now, the problem with this type of approach is it does answer the question, but it only answers the question for a single T cell receptor. And we really need to be able to move further than a single T cell receptor if we're trying to make conclusions about what is a part of a normal repertoire. So to do this, we needed a way to multiplex this type of assay for in vivo analysis. And for this, we used the system called flow codes, which is a modification of the ProCode technology generated by Brian Brown. Now, this technology was published a few years ago in Cell by Brian's lab. And essentially what it does is it uses 14 different antigen tags that have all got available antibodies. So here you can think of things like a HA tag or a V5 tag. Now, those 14 tags by themselves would just give you the ability to multiplex 14 different barcodes. But the really insightful aspect of this ProCode paper is that the Brown lab created linear epitopes of 3 antigen combinations. Now, once you get into combinations, you can get 364 unique combinations of tags based on those 14 different antigen tags. And you can put each one of those combinations into a lentivirus and give it a cargo that will be unique per lentivirus. And then you can use this to multiplex an experiment. Now, this was validated in vitro with CyTOF technology in the Cell paper. Unfortunately, there are limitations when it comes to using this system in vivo and with flow cytometry. So in order to overcome those limitations, we developed the flow code version. In this, we use the same antigen tag combos as the ProCode library, but we overcome some of the limitations by fusing these ProCodes to histone 2b. Now, the reason we do this is that the intracellular tag localization prevents rejection of the cells when you use them in vivo. The tag also builds up to much better levels for detection by flow cytometry and allows us to stain intracellularly for things like FOXP3 without losing the tag. And we've now validated this process in vivo with flow cytometry. This system was really developed in our lab by Adrian and Magda. And Adrian also developed the necessary software in order to debarcode all of these barcoded cells. This is Flowcode Decoder, and you can access the software through the QR code. Essentially what that does is it automatically gates positive negative populations for all of these tags and then assigns which combination tag the tag results from on a per cell basis. So this is how it works in practice. We have a set of retroviruses, and each of those retroviruses has a cargo, which in this experiment will be a TCR alpha beta chain, and then it also has a histone 2b fusion. And if we look at multiple different cargoes, multiple different retroviruses, each of these will have a different cargo and a different known fusion protein, so that we can use flow cytometry detecting all of these different tags and then using that to assign the cell to one of the different barcodes. Therefore, we'll know for each cell which cargo it had. Does this cell have TCR pair 1, TCR pair 2, TCR pair 120, so forth. This is how the experiment is designed. We have to clone all of these retroviruses, clone in the tag, clone in the TCR combination. We then produce virus and put them on hematopoietic stem cells, and then we can transfer those hematopoietic stem cells into a radiated mouse, so they develop into T cells that are each carrying one of these different TCRs. Now, the experiment I'm going to show you now is a proof of concept with 20 different retrogenics that are traced by flow cytometry, although we are now doing experiments where we can trace hundreds of these simultaneously in the same mouse. Now, this is the type of data that we get. This is quite a complicated slide, but I'll go over it in just a little bit of detail. Now, what we've got is the different TCRs that we cloned from regulatory T cells, and all of these TCRs are now being expressed in our T cells in our mouse, and we can track each of these clones individually with the barcode. The circles are looking at and the top three circles, the size of the population. Size of the circle is how big the population is, and then the color of the population is whether or not they are regulatory T cells. If they're blue, they're not regulatory T cells. If they're green, they're regulatory T cells at a pretty physiological rate. And if they're yellow, orange, or red, they're regulatory T cells at a super physiological rate. And what you can see automatically straight up is that there's a lot of green and yellow in there because these cells are almost all becoming regulatory T cells. You can compare that to the first column of OT2 cells, which are not becoming regulatory T cells in the lymphoid tissues or in the non-lymphoid tissues. They do become regulatory T cells a little bit in the gut because the TCR here recognizes an egg antigen and there is some egg in the food of the mice. If we look at the other 20 TCRs, we see that they're able to become regulatory T cells across most tissues. Not every regulatory T cell not every TCR drives regulatory T cell fate in every tissue, but we see that for most of these, there is a distribution of regulatory T cells across multiple different tissues. There are a number of these clones, and you can see them with the asterisks at the bottom. For example, Treg clone 12, which was originally extracted only from the kidney, and we find that that particular TCR drives a very kidney-skewed population of residency. So that would be an example of a TCR that drives a kidney-biased outcome, whereas something like TCR7 gives a very balanced regulatory T cell outcome with high representation across multiple different tissues. So if we go back to this idea of can we measure whether or not the TCR that we clone drives a Treg fate? Yes, 20 out of 20 of these TCRs drove Treg fate. And then are they becoming monotissue or multi-tissue in their homing? Now, all 20 of them are multi-tissue, but 5 of them have bias to the tissue of original detection. So I think what we have here is many Tregs that are quite tissue agnostic and some Tregs that are tissue biased, but at least in our small set, we don't have any Tregs that are really tissue exclusive. Now, the third test of this pan-tissue model is that regulatory T cells from tissues, if they really are pan-tissue in nature, they should be tissue agnostic on rehoming. Now, for this, we had an experiment from Oliver Burton where we extracted Tregs from different tissues and then transferred them into a new mouse and looked where they homed. The way we did this is to have multiple different congenic strains. So we can have CD45.1, CD45.2, CD45.1.2, CD45.1 GFP, and so forth. We can then take organs from each of these mice, take multiple organs, and then create a pool where each organ comes from a different mouse. So in pool A, we might have the liver from CD45.1, the lung from CD45.2, and the pancreas from CD45.1.2. So this creates multiple different pools, and in each pool, we've got regulatory T cells that we can trace which tissue it originally came from. We then transfer the entire pool over to a rag knockout mouse and look where those regulatory T cells home after transfer. We can calculate a migration index, which is essentially working out, are the regulatory T cells more likely to go back into their original organ, in which case they're going to have a high migration index, or are they tissue agnostic, in which case their migration index will be 1, or are they actively avoiding the tissue they came from, in which case their migration index will be less than 1. Now, what we see is that gut Tregs do have a bit of a bias towards going back to the gut. They're about 2-fold more likely to go into the gut than they are to go into another tissue. But if we look at kidney Tregs or liver Tregs or lung Tregs, they are completely agnostic. They're just as likely to go into a different organ as they are to go into the original organ that they were harvested from. So if we're taking a broad look at this new data, I think it does support the pan-tissue model for tissue Tregs, in that we've got circular migration, we've got a generic tissue residency program, which is implemented more or less the same program across multiple different tissues, and we have quite transient residency on the order of weeks to months. Now, we do think there is a distinct gut population, and there are probably also some TCR specificities that have got a more restricted migration pattern than the general tissue Treg. Now, just before I finish up, though, I want to move on to some unpublished data on asking how these tissue Tregs enter the tissues. And this is work from a very talented student in the lab just writing up her thesis, Magda Ali. And what she asked was whether or not we could calculate what molecules were being used by regulatory T cells to enter each of the different tissues. Now, there's a relatively discrete set of migration molecules that are available for the tissue entry. There are integrins, adhesions, cohesions, selectins, chemokine receptors, kinetic receptors, and so forth. But all up, that's maybe 150 genes. And so what we could do is clone the guide RNAs from each of those genes into a flow code retrovirus and use Cas9 to knock out each of those 150 genes, and in a mixture of cells. We can then transfer that mixture of cells into a RAG knockout mouse and see if knocking out one of those genes prevents that particular cell from entering a particular tissue, but multiplex it, where we can look at all 150 guides in a small number of mice. And then we use spectral flow cytometry to measure the frequency of each barcode in different samples. So this is the type of data that we can get out. This is looking at 10 different mice. So we can analyze in 10 mice 155 different knockouts simultaneously, and we can do this in 18 different tissues. So this is 180 FCS files, which is actually remarkably low considering the number of different knockouts we're able to analyze in parallel across 18 different tissues. And so for each of these 155 guide RNAs, which are knocking out one gene each, we can ask what impact do they have on the frequency of regulatory T cells within each particular tissue. And this is simply by looking at the frequency of the code being detected between two samples, say the spleen and the lung. We can also ask questions like, do different subsets use different molecules? We can look at CD4 T cells, CD8 T cells, Tregs, all in the same experiment. We just gate a different population and then look at the barcode frequency within that population. And we can ask things like, with knocking out a particular gene, changes the phenotype. We can do a t-SNE of the knockout or wild type cells. We can look at the ratio of wild type to knockout barcodes in naive cells or activated cells or memory cells. And we can do all of this analysis from the same experiment because it's all based on flow cytometry. So this is a heat map looking at 150 different genes, what impact each of those genes has got on regulatory T cells entering each of the different tissues. It's quite preliminary at this stage, but I can show a few types of examples that we'll get. So this is looking at regulatory T cells entering the lymph node. So it's a volcano plot, type of volcano plot you'll often see for expression, but this is not expression, this is a functional analysis. So which genes impact the ability of a Treg to enter the lymph node versus enter the spleen. And it's actually quite limited in terms of what genes are important. There's two integrins, ITGAL and ITGB2, which are very important for cells, for Tregs to enter the lymph nodes. And then on the spleen side, we see S1PR1, which I think is actually not important for entering the spleen, but it's rather important for leaving the tissues. It's a retention molecule. And then we can zoom in on that SP1R1 and look at the impact that losing that gene has got on entry into multiple different tissues, for example. We could also compare the gut tissue to the spleen. And here's an example of what's important for regulatory T cells to get into the gut versus regulatory T cells to get into the spleen. Now, if we look at the gut entry, it's really a single integrin that seems to dominate the effect here, ITGA4. And we can see that the cells that have got a knockout of ITGA4 are quite present in the spleen. They're present in regular lymph nodes, or the axillary lymph node, but they're a bit lower in the mesenteric lymph node that gains the gut, and they're much lower in the Peyer's patches, the LPL, and the IEL. So this is a gene that is very important for entering the gut. But you see, there are no other genes that have a very high importance for entering the gut. So it's a relatively simple migration system into this tissue. We could go through every tissue one at a time, but the data set is still quite preliminary, and hopefully we can release this soon. So just to sum up the system of flow codes, these are a really useful technology for doing massively parallel in vivo experiments. Now, it's single cell barcode, but the great thing is that we can read out by flow cytometry. This means it becomes cheap and fast to get data on millions and millions of cells. Compared to something like Perturb-Seq, where you can spend your entire lab's budget on a few thousand cells, this gives you really a unique opportunity to do highly parallel screening. The other thing that's really useful is got very high recovery because the readout is flow cytometry and not sequencing, so it can be very good for rare populations such as tissue resident cells. We can either barcode in CRISPR guides if we want to create knockout libraries, or we can barcode in mRNAs if we want to look at the effect of genes, for example, tracking hundreds of TCR clones simultaneously or hundreds of antibody clones simultaneously. It's also compatible for image stream, so you could screen for subcellular localization, and it's compatible with intracellular readouts such as FOXP3 expression or switching on a cytokine. Now, there are limitations to it, and at the moment, with the current depth, the limit is 365 barcodes per library, although of course you can cover multiple different libraries by simply creating multiple libraries and running them in different mice. The other big impact is that new libraries really require individual cloning of each set of retroviruses. So it's a lot more work to create a library using the flow code system or ProCode system than it is to do mass cloning for a sequencing-based readout. And finally, it requires access to either spectral flow cytometry or to CytOff. You don't really have the ability to do this on a standard cytometer because you're going to end up using 14 of your channels just to read the codes, and you need to have more channels to read those phenotypes. Now, if you would like some further information on any of our flow cytometry techniques, our guru is Oliver Burton. You can contact him via his email. I'd really suggest, though, looking at his blog, Colibri Cytometry. There are hints and tips for ultra-high parameter flow cytometry, all the protocols he's developed, all the common problems in flow cytometry. He's tackled them and put them on that blog. There's also some key cytometry resources developed from the lab: flow cytoscript, flow code decoder, auto-spill, and overnight staining protocols. And with that, I'd like to finish up. I really want to acknowledge the fantastic people in our lab, in particular, Oliver Burton, Oriane Bricard, Magda Ali, and James Dooley for this project that I presented today. Key collaborator, Brian Brown's lab with the ProCode technology. If you're interested in any of our data sets, you can use that QR code. And we've also got a set of articles on career advice for any junior researchers looking at a career in academia that you can get on the bottom barcode. Thank you very much.
So we will conclude the webinar there. Thank you everybody for attending. Thank you, Professor Liston, for your really wonderful talk, and great morning, afternoon, or evening wherever you are in the world. Take care.
Thank you.